Good morning. So today Wenjin will be introducing Capybara, a computational tool to measure cell identity and fate transitions. So over to you, Wenjin.
Thank you. Okay, let me share my screen. Okay, can everyone see my screen? Okay, so today I will be talking about Capybara, a computational tool that we have developed to measure cell identity and fate transition. So today I will be focusing on the method and the pipeline of Capybara more instead of the results. So hopefully I can clearly explain what we actually did in this computational tool. So in Capybara, we in general have 3 different steps. The first step involves tissue level classification, and the second step involves a construction of high resolution reference and a continuous identity measure. And the third step, we perform a discrete cell type classification and multiple identity cell scoring. So I will go through each step in details. Let me know if you have any questions in these processes. So in the first, before I get into the pipeline, I want to again describe this idea of quadratic programming. So quadratic programming is takes the assumption that each transcriptome of a single cell is a linear combination of the bulk transcriptome of different cell types, with these each fractional identity labeling how likely does each single cell belonging to a cell type. So where these fractions are taking the restriction of that, the sum of these fractions will add up to be less than or equal to 1, as well as each of these fractional identity has to be greater than or equal to 0. So this approach has previously been used in different methods, particularly involving reprogramming, where here in 2016, where in the Barbara Chulin first proposed using this method in the paper to measure the identity of reprogramming from mouse embryonic fibroblasts to neurons. And it is able to measure this gradual transition from the starting population to the terminal population. And back in 2018, we were able to also use this method in our reprogramming system from fibroblasts to induced endoderm progenitors, where the 2 cell fate that we're measuring here is the fibroblast and the endoderm progenitor. Further, we were able to develop this tool to take single cell RNA-seq references and perform analysis to further detailed classify these single cell RNA sequencing data into different cell types using a reference that is building from a single cell atlas. So this will be the building block of Capybara, and I will be referring it as QP in the description. Follow along. So in the first step, we are going to do a tissue level classification where we are trying to identify the tissues that are associated with our single cell RNA-seq sample. So why is this classification of tissue necessary? So the reason why we need this is to reduce excessive dependencies caused by the correlation across tissues in the final high resolution reference. So take an example here that if we have an actual cell type, for example, a lung fibroblast in our single cell sample, however, in our in different tissues, they have a different type of fibroblasts. If we just pull all of these tissue together, we will get a result that the fibroblasts from different tissues will all receive a relatively high score. So using the tissue level classification to remove the tissues that are not relevant to the single cell sample would help reduce this issue so that we will not even include this in the further downstream analysis. So the goal of this step is that if we have a single cell expression profile, which is our sample, and then we have our annotated single cell atlas, we are trying to ask the question, which tissue in the atlas best capture the sample cells? So take the example here that we use the mouse cell atlas, and then our data, which is what we used in the paper back in 2018 of the reprogramming from mouse embryonic fibroblasts to IEPs. The goal is to identify these these tissues inside of the mouse cell atlas that actually maps to our single cell RNA sequencing data. So to do this, we are bridging the single cell atlas data and the sample single cell RNA-seq data using the bulk transcriptome. So the bulk transcriptome, we were able to mine from this platform called ARCHS4. So they did a massive parallel mining of public available RNA-seq data from GEO. So they include a lot of different samples from human and mice, also including a lot of ChIP-seq data. So it is a very valuable resource. And then we take only the poly-A and total RNA-seq data from the mouse with strain of C57 black 6 mouse. And we were able to construct a final reference with 30 different tissues across the mice. So this bulk reference is also available along with our Capybara package in GitHub. So using this bulk reference, we will be able to run quadratic programming through using this reference cell atlas, from which we will get a fractional identity matrix of each individual cell mapping to the bulk transcriptome tissue. Along with using the single cell atlas, we will also run through quadratic programming using our sample single cell RNA sequencing data with this bulk reference, which we will get for each different sample cell, we will get a score also for these different tissue types. So using these two fractional identity matrix, we will have the reference QP score matrix, which is the fractional identity matrix of mouse cells, let's say here. And then we will have our sample quadratic programming score matrix. And we will calculate a cell-by-cell correlation based on these fractional identity score. So the idea here is if they share the similar pattern, the sample cell should map to this tissue type inside of mouse cell atlas. Using this correlation here, as we can see, we will be able to select 3 different tissue types from this comparison, which involves lung, liver, and bladder. So taking from there, we will construct a high resolution reference and then perform the second step, which is to do a continuous identity measure on this high resolution reference. So starting from the selected tissues, we will go back to the mouse cell atlas again, which will give us the different expression profile of the single cells of each cell type. And we will take a liver as an example here. From each cell type of the liver, we will select reference n different cells. So the default here is 90 cells. So we choose 90 cells based on previous evaluation on different cell number for performance. And then in these n different reference cells, we will choose half of them to be the most homogeneous cells and half as the most diverse cells. So using this, we will calculate an average of these expression profile to build a final pseudo bulk of this cell type for each cell type inside of liver. So using this, we will be able to construct a high resolution reference including all the different individual cell types that are involved in this single cell RNA sequencing sample. And then using quadratic programming again, we will be able to get the fractional identity matrix for each cell in the sample data set with the cell type identified in the high resolution reference. Moving forward, using this continuous identity score measuring, we are going to perform discrete cell type classification and multiple identity scoring. So along with that continuous identity measure in the sample, we also perform a continuous identity measure in the reference. So here, this on the left-hand side is what we identified as the expression profile of each single cell of each cell type from the mouse cell atlas. And then we will also run through quadratic programming using our constructed high resolution reference. And from this, we will get a fractional identity matrix for each cell in the reference data set, which will serve as a background in this classification. So based on this background, we will further perform empirical p-value calculation. So in this case, on the top here, we have the fractional identity that we observed from the background. On the bottom here, we have the fractional identity we obtained for the sample RNA-seq data. So from the top here, we will be able to construct a distribution of these fractional identity scores for each cell type. And then building from these distributions, we will be able to see where does the fractional identity of the sample falls into this distribution, where we further calculate this empirical p-value to see what is the likelihood of getting this such a high or low score. So so basically, we will project this and we sample this for a total of 50 times so that we get 50 different empirical p-values, and we can kind of measure this p-value in a distribution as well. So based on this, we will get an empirical p-value matrix. So here, this is the reference empirical p-value matrix. And then because for the reference data, we already know what is the cell type of each reference cells, we can use these empirical p-values to obtain a benchmark empirical p-value. Using this benchmark empirical p-value for each sample cell, we will be able to get a cell type binarized cell type call, based on which we will be able to call their cell types accordingly, and where when the cells have more than one identity called, we will label them as multiple identity cells. So the last part of demonstrating the method is to discuss about the transition score, which is a development from the multiple identity cells. So here I will explain the transition score in this model here, where each cell state is a discrete cell type, and the cells that are connecting the cell states are the cells that having multiple identity going to the two different cell states. So we are interpreting the quadratic programming score as the transition probability here, that how likely is one cell going to transition into one cell state. And then the transition score here is actually measuring each cell state instead of each cells. So consider the one cell state in the following, for example, the cell state 1 here, based on in the information theory, the information of each transition is equal to the negative log of each transition probability. And the information received by from each transition of the cell state, it can be calculated by multiplying the information of the transition by the p-value. And so that the total information received by each cell state can be represented by the sum of the transitions that are entering or leaving the cell state. So using this transition score measure, we were able to actually see how what is the flux and like how dynamic is each cell state. And kind of measure the transition from there. So that is the end of the methods part. Next, I will go through the tutorials on our GitHub page to briefly explain what are some of the key steps in running Capybara pipeline.
Thank you, Wenjin. That was really nice. It's very concise. I liked it. So I have a question about the references and the transition states. So what if your single cell reference is particularly sparse, or there's a major cell type missing from that reference? Then how will the transition scores, how will they look under those circumstances, and what should users be aware of?
We are talking about single cell references being really sparse. So hopefully that issue can be resolved when we are constructing the pseudobulk, because a reason why we want to construct the pseudobulk is because single cell RNA sequencing data is very sparse and it has the issue of dropout. So considering the selecting the reference cells from the single cell RNA sequencing data, it should be able to get enough information from across different genes, and also like construct a relatively less sparse reference. Additionally, the reason why we consider both the most homogeneous population and the most diverse population in the same cell type is because so that we capture the variability in the gene expression, so that it contains all the genes that is expressed and inside of each cell type. So hopefully in the reference construction part that will be able to help resolve that issue. And then regarding if that cell type, if there is a missing cell type in the reference and that is not mapping to the single cell, there are, so most likely we would hope is that they will go to the unassigned category so that they are not assigned to any category of our cell types in the reference. However, if it has any relevance to any cell type in the reference, it might be able to be characterized as one type of like a multiple identity cells. However, it might like share really diverse cell types across the board. So, yep, I hope that answers the question.
It did, thank you. Any other questions?
Yeah, I have one. Also about the reference genome. What if you don't have a ton of, what if there's a lot of overlap between the cell types in your reference? So what if, because I'm just thinking about how quadratic programming works with this idea about linear combinations of all of your discrete cell types. What if there is not a lot of discreteness? I don't know what.
Yeah.
Does that make sense?
Yes. So that is another reason why we perform tissue level classification first, because in different tissues, they share a lot of the same cell types. So quadratic programming, once before, I kind of validated that it can deal with some of the interdependence between the two different cell types. However, like when the dependencies gets like really big, it will be a problem because of that the reference itself is not independent from each other, like they are not different enough anymore. However, using the tissue level classification, we are able to really bring the cell type down to like the significant, like so that there will be some overlap. However, like it will not, like it will still have significant difference between the different cell types across tissues. Yes, so one way that I describe the tissue level classification is we're selecting for the tissue level features first, and then we select for the different cell type features of the of the transcriptome space. So hopefully in that 2 steps, there will be able to remove a lot of the interdependencies between the different cell types.
Can I ask a question about the tissue level specification?
Yeah.
So, will your script always give us the top 3 most likely tissue, or should we draw a threshold to cut off the score?
Yeah, so that's a good question. And so the threshold right now is very dynamic. So you can choose it on yourself. So right now, I did provide a threshold that I have used basically for the dataset analysis. It's not just like the top 3. It will involve like how many cells are getting captured through this reference. So basically, we are hoping that we will be able to capture most of the cells in the sample RNA sequencing data in this tissue level classification. So based on that, we will be able to draw a threshold. Also, this will depend on the cell number that we are having in the single cell RNA sequencing sample. For example, if we have 85,000 cells in like our reprogramming data set, then the threshold will be drawn like maybe a little bit higher or lower, considering like what percentage of cells do you want to include in your classification. So it can be very dynamic at this point right now. Okay.
Any other questions? Okay. Thank you, Wenjin.
Okay.